Okay, um, so we are on slide number 21, and we are discussing the interrupted gene, right? So the first intron is usually the same size for a family of genes. Notice that I said two words that are important. One, usually, not always, and the second one is for a family of genes. What is a family of genes? A family of genes are more than one gene that have something to do with the same process in the body or the same trait. So for instance, we can see genes that have something to do with metabolic abilities, okay? Just for instance. Families of genes can also be related between species. So we can compare the same gene with its family in mice, in rats, in pigs, in cows, and humans. All right, for instance. The second intron usually is what we would call a difference maker, and it has various of sizes in the between families or within the family. When the right exon intron order is kept between species, the length of the genes vary due to the differences in introns. So just remember that we can have differences in the order, in the sequences of exon introns, and also in the length. Okay? Alright, so what we can see on in slide 22 are three examples that I made up, completely made up, for three different species. So let's start with the upper one, and you can see on the left upper corner that we're writing a gene of species number 1. So it has 3 exons, right? 1, 2, and 3, and it has 2 introns. And then we're going to compare that to species number 2. So we see that even if the exons are the same, and I made them the same on purpose, and you can, and how do you know that they're the same size? Because underneath every intron and every exon, I wrote you how long is the sequence, right? So for instance, exon 1 is 600 BPs. Exon 2 is 650 BPs, and exon 3 is 800 BPs. I'm just making it up. It doesn't really matter the number right now. But we can see that between species number 1 and number 2, for instance, intron number 1 for species number 1 is 650 BPs, right? But the intron number 1 for the second species is only 550. And you can see how I just made up numbers and played and created 3 different lengths of the gene. Okay, so it's just an example and you can play with it however you want. Genes that are related by evolution are organized in a similar way. So usually the position of the introns is going to be a major thing that is similar. Okay, and also the exons. So the level of similarity between genes depends on 2 major factors. 1, the final function in protein, right? That's number 1. And 2, the origin of the genes, meaning that it depends on the duplication of an ancestral gene. Okay? So basically when we want to find families or correlate between genes, we're going to want to find that ancestral gene that and start working up the chain, so to speak, working up through evolution and finding the links. And 2 major ways to do that is the introns and the exons, right? Of course, the protein that is produced from the coding sequences, right? Okay, so we are on slide 24. Of course, with time, meaning through evolution, and evolution is something of millions of years, the genes accumulated changes. So the more duplications of a gene in the species and between species have been made, the more chances there are that we will find changes. And those changes accumulate, and then you have branches, right, just branching off the ancestral gene. So the exons are actually the building blocks that are assembled in various combinations, but we find them to be relatively highly conserved between families of genes. Once again, they're not identical, but they're highly conserved, especially essential exons or components of the gene. Okay, let's move on to slide number 25. So basically, two genes may have identical exons and look completely different because of their introns. Okay? Mutations occur at the same rate in both introns and exons. So it's not that we can say that one component is weaker or more susceptible to mutations. Okay, but they are removed by what we call adverse selection from the exons, meaning that since the exons are the ones that code most likely for protein, right, they're going to be addressed in a more severe way. Let's see. Okay, so one question can be, okay, introns accumulate mutations, right? But they don't affect the outcome, most likely, especially not the protein. So are they at all needed? So here's the thing. On the one hand, we're playing here, so to speak, with evolution, right? Evolution takes millions of years. So let's say that a change is going to occur, right? Some kind of change is going to happen in the future. And let's say it's going to take 1,000,000 years for it to happen. 1,000,000 years is a very doable, let's say, logical time span when we're talking to evolution, all right? It's actually kind of short, if you ask me, in most cases. But let's say it's going to take a short time, just 1,000,000 years for it to happen. But let's say that the clock started in 2010. That means that right now a process has begun. Theoretically, we know about it. We know it's going to take 1,000,000 years, but it's only 2015 right now. So we're only 5 years out of 1,000,000. So when we take my genes and your genes and we look at the introns, we're going to say, what is the importance of the introns? But in 1,000,000 years, something's going to happen to those introns and going to change. And we're either going to have an advantage gained or a disadvantage, okay, because I defined that something is going to happen, right? So we eliminated the option of nothing changing. But we're only 5 years into the process. So right now, when we look at the genes, it seems as if the introns are not important. But who says that in the process of evolution, in this game, so to speak, of evolution, that introns might not have a role to play. The second option to say is, hey, so much thought has been put into DNA. It's so brilliantly designed. So many years of evolution. Some kind of guiding force. Why weren't they eliminated if they have no role to play? Okay? So, on the one hand, we can't prove. On the second hand, there's some kind of logic to it. I believe in the first idea. They're there for a reason, and since it takes millions of years to find out why, at any given point, it's really hard for us to see the whole picture. We're just seeing really, really small fragments of the bigger picture. So let's say you are a researcher, okay, and you dedicate your life to genetics, and you're researching the role of introns. And let's say that you started at the age of 20 and you die at the age of 80, and you've never went to a restaurant and you've never went out. You don't have a family, you don't have a life. All you do is stay in your lab and research. 60 years you sleep in the lab. That sounds like a lot to us. But now take those 60 years and divide them by 20 million years that a phenomenon might take to occur, if at all. And what did you get? Nothing. It's like a water drop in the ocean. But if you do not accumulate those drops, you don't have an ocean. Alright, so that's very important. So it's really hard to understand, in some cases, the role of certain components, because it might take quite a long time to see change. Alright? So we need to remember that. Every time that the presentation talks about evolution or mentions it, or I say evolution, remember we're playing in a game that takes millions of years. Imagine a football game and not even one touchdown is scored. What are you going to say about that game at the end? That it was disappointing, right? Right? Or a basketball game that not even 1 point is scored, or a baseball game, or any game, any sport. There's no score, and we're really disappointed. This game, so to speak, of evolution takes millions of years. So it's hard for us to grasp what's going on. Okay, let's move on to slide 26. You can see that between species we find conserved genes that have always an ORF, right? And have a related sequence. That's why they're called conserved. Of course, such genes have to have an essential role, otherwise they're usually not conserved, especially not between species, right? But these two features of conserved genes allow us to isolate certain genes and link them one to each other. Okay, so the more similar two genes are, we can correlate them and understand what they are about. So the less introns that a gene has, the less interrupted it is. In regards to slide 27. But here's the thing. Introns kind of make it harder to correlate between genes, genomes, the size of the organism, and the size of the genome, right? They kind of make us go lost because theoretically we don't need them, but they vary in size and numbers and positions. So they're kind of, shifting our focus and concentration from the parts that really code for protein. So they kind of, interfere a bit in a way. Once again, it doesn't mean that they don't have a role, but when you're trying to compare two genes, the introns are kind of disrupting your concentration in a way. So in higher organism, exons code for smaller stretches of protein of about 50 amino acids. That's one of the characteristics of higher organism. And during evolution, small protein coding units have been added to the human genome. So when we map backwards and start correlating between genes, we see that those units have been added. Okay, we'll stop here and continue from slide 29.